Hello student, good morning to one and all. I am Dr. Tariq, Department of Biotechnology, Islamic College, Manembadi. Today, before we discussed about bioinformatics topic on Swiss Switzerland protein database. So now our topic is this hour we discuss about CSD, that means Cambridge Structural Database. I asked asking the question for part A, what is meant by CSD in bioinformatics? So you write down the abbreviation of Cambridge Structural Databases. Okay, so this topic is being discussed in this junction. The Cambridge Structural Database, that means CSD, is a highly curated and a comprehensive resources. It's established in 1965. The Cambridge Structural Database is the world repository for small molecule organic and metal organic crystal structure. The CSD, Cambridge Structural Database, the world repository of small molecule crystal structure. That means the CSD records bibliographic, that means reference, chemical and also crystallographic information for organic molecules and metal organic compounds. Actually, whose 3D structures have been determined using X-ray refraction and neuron diffraction. X-ray diffraction and neuron diffraction. And CSD records the result single crystal studies and powder diffraction studies. So these are all the main field of the CSD data deposits. Now the structural bioinformatics databases. The databases store the 3D structure of information obtained the experimental techniques, particularly in crystallography, NMR, electron microscopy of proteins, DNA, RNA, viruses, carbohydrates, lipids, and others someone. Actually, the protein data bank PDB, primer molecular macromolecular structure database. The protein databases is the official website ebiac.uk. The structural classification of proteins is official website is cop.mrc-lmp.cam.ac.uk. Finally, CATH databases as official websites are the Cambridge Structural Database, that means CSD. That contain 3D structural information of small molecules. The structure databases of the CSD, the biology protein structure database is the database that is modeled around the various experimentally determined protein structure. The aim of the most protein structure database is to organize and annotate the protein structure. It's providing the biological community access to the experiment data in a useful way. The CSD means Cambridge System Database, Structural Databases. CCDC means Cambridge Crystallographic Data Center. Now, the crystal structure is in electron microscopically crystal structure description of ordered arrangement of atoms ion molecule in their crystalline material. The crystallography is the word it derives from the Greek word crystalline, cold drop, frozen drop. This means that all solids some degree transparency and graphene. The actual official and websites means CSD, Cambridge Structure Databases. The basically we discussed in the previous slide. That means CSD official websites, 3D structure. These are the 3D structures. The 3D bond will attach to the particular target DNA base databases. It's combined together. It's automatically new drug will be displayed. The lot of analysis history list is there inside line. Be suitable for each and everyone should join the bond. It will be adjacent and equal to the joint in the one to another pair. So exactly it will be merged the bond. The drug will be successfully creation in the easily method. So these are all the CCDC Cambridge Crystallographic Database Center. The official website is there. So 3D visualizer, we selected the 3D visualizer, explanation are there, author, text, diagram, CSD internal, everything will present in this reference. Okay, these are the basic applications, softwares are available in this system, right? The crystallography is the experiment science determining the arrangement of atoms and the crystalline solids. That means crystal structure, X-ray, Crayon loop, and a detector. The finally this one is present. Okay, so these are all the 3D structure in the every structure. Then CCDC means Cambridge Crystallographic Data Center. It's the home of small molecule crystallographic data and a leader in this software, pharmaceutical discovery, materials development, and research and education. It is originating the Department of Chemistry and University of Cambridge, and it is non-profit organization whose primary role in the compilation and maintenance of the CSD, that means Cambridge Structural Database. They also perform analysis on the database form for the benefit of the scientific community. Then Cambridge Structural Database, that means a CSD, it's a database such as a repository and the validated the curate and the resource for the three-dimensional structure data. That means the molecules or crystal structure hosted by CCDC. Actually, it is stored bibliographic, that means reference, chemical oriented, and the crystallography information. The organic molecule and the metal organic and organometallic molecules are present. The specific entries of the complementary to the other crystallographic database such as the protein data bank PDB, inorganic crystal structure database in International Center for Diffraction data. The data typically obtained by X-ray crystallography is less frequent by neuron diffraction. The content is CSD Cambridge Structural Database is updated with about 50,000 new structures each year, and the improvement to existing entries that means structure is repository or related for public access shortly after the corresponding entry has appeared in the peer-reviewed scientific literature. The authentication of the content is CSD. The in side view, that means right side there, the each and every protein structures will authenticated, and as well as the structural databases and how much percentage in each and every species. So these are all the side view of the content. The another one is the official websites are CSD, that means Cambridge Structural Database. These are the actual websites present in the various information, particularly in the references and the depositor, the analysis. So everything we present, we and discuss in the clearly and notably informations, particularly in references we collect. After that, your research is going on the data. Okay, the CSD community. The community also is important for the crystallographic tools, particularly in data collection, validation, and visualization, to teaching, research, and analysis. These are all the analysis in the tools in the CSD. Finally, the CSD teaching databases. CSD teaching databases almost about 300 structure plus subsets carefully selected then to enhance chemistry learning. The picture shows the 3D structural. These are all the illustrate fundamental concept of bonding and structure. The conformational analysis, stereochemistry, and the reaction mechanism. Finally, the examples of the all major chemical functional groups, as well as a wide range of broader chemical classes. It includes carbohydrates, nucleotides, amino acids, steroids, porphyrins, alkaloids, structures, and finally the high polymers. These are all the compound joined together the very broadly and the functional groups in this CSD classes. So thanks to watching in this class. Thank you.